Welcome to our deep dive into DNA replication, the process that lets life copy itself flawlessly into billions of times a day. Without replication, there would be no growth, no repair, and no second chances for your skin cells after a sunburn. In this video, we will explore how enzymes like helicase and DNA polymerase make it all happen. Don't worry, by the end, this won't be just memorization. It'll actually make sense. Let's unzip some DNA and get started. DNA replication is the biological photocopying machine that ensures that every new cell gets a complete instruction manual. It happens during the S phase of the cell cycle inside the nucleus. Every strand of DNA serves as a template for building a new complementary strand. The result? Two identical DNA molecules, each ready to guide a brand new cell. Nature, it turns out, was into backups long before humans invented cloud storage. The process begins when helicase arrives on the scene. Think of helicase as the unzipping enzyme. It breaks the hydrogen bonds between base pairs, separating the double helix into two single strands. The point where the splitting happens is called the replication fork. Now we have open strands that can act as templates. It's like unzipping a sleeping bag, except way more precise and with fewer regrets. Next up is primase, the underrated sidekick in the replication story. DNA polymerase can't start a new strand on its own. It needs a short RNA primer to show where to begin. Primase lays these primers on both strands. Without this step, polymerase would just sit there, staring awkwardly at single-stranded DNA, unsure of what to do. Every superhero needs a sidekick, and Primase plays that role perfectly. Now, the star of the show enters, DNA polymerase. It reads the template strand and adds complementary nucleotides. A pairs with T and C pairs with G. The enzyme can only work in one direction, from the 5' to the 3' end. That directional rule will matter a lot when we get to the lagging strand. DNA polymerase is a perfectionist. It even proofreads as it goes, catching most typos before they become permanent mutations. Because DNA strands run in opposite directions, replication isn't symmetrical. On one strand, DNA polymerase can move smoothly and continuously, the leading strand. On the other, it must work in chunks, the lagging strand, creating short pieces called Okazaki fragments. These will later be glued together, but for now, just remember, DNA replication is efficient, not perfectly elegant. Like assembling IKEA furniture, it gets the job done. Once the lagging strands fragments are in place, DNA ligase comes in to do some molecular glue work. It connects the Okazaki fragments by sealing the sugar phosphate backbone. Without ligase, your DNA would look like a broken necklace. Technically all there, but falling apart. It's the finishing touch that gives you two complete, continuous DNA molecules ready for cell division. DNA replication follows the semi-conservative model. Each new DNA molecule contains one original template strand and one newly synthesized strand. This design makes replication both stable and accurate. It's like copying an essay while keeping one original paragraph to double check for mistakes. Semi-conservation isn't just efficient, it's genius molecular engineering. DNA polymerase doesn't just build, it also edits. As it adds bases, it double checks that every pairing is correct. If it spots an error, it removes and replaces the wrong nucleotide before moving on. This proofreading ability keeps mutation rates incredibly low, roughly one mistake per million bases copied. Humans could take a few notes from that kind of accuracy. DNA replication underpins everything from cell division to evolution. When you heal from a cut, replication makes new cells. When organisms reproduce, replication ensures offspring inherit complete genetic blueprints. Mistakes in replication can cause mutations, some harmful and others beneficial. In medicine, understanding replication helps explain cancer and guides genetic therapies. So, yes, this topic is both exam gold and real life crucial. On exams, students often lose points by forgetting enzyme names or labeling strand directions incorrectly. Always specify the role of helicase, polymerase, primase, and ligase. If you see a diagram, label 5' and 3' ends, replication forks, and fragments. And never forget to mention semi-conservative replication. It's one of those phrases that makes exam graders smile. That's it for now. Don't forget to subscribe, like, and check out Dr. B's tutorials on Amazon for full AP and IB study guides. See the first link in the comment below. For more tutorials and test prep videos, subscribe to our YouTube channel, Dr. B's Tutorials.